Yesterday, we talked about mitosis, a type of asexual cell division. And if you would look in your body, we said, in your body, all the cells except either the eggs or the sperm were made by mitosis. Eggs and sperm aren't made by mitosis, though, or if we were looking at a plant, pollen, or if we're looking at some types of organisms, spores. So if we look at those cells are all made by sexual cell reproduction, and they're all made by a process called meiosis. Unfortunately, meiosis and mitosis sound a lot alike. But in meiosis, we start out with a cell that's diploid, that has the 2n number of chromosomes. So it has 2 copies of number 1, 2 copies of number 2, all the way down to 2 copies of the sex chromosomes. And at the end of meiosis, we end up with cells that are 1n, that are haploid, that only have 1 copy of each pair of homologous chromosomes. And then these cells are used to reproduce the next generation. Think of the egg and the sperm. Spores and pollen do similar functions. So when we look at meiosis, the easiest way to think about the difference between mitosis and meiosis is, in mitosis, the cell replicates its DNA. So after it replicates its DNA in mitosis, temporarily, the cell has twice as much DNA as normal. Then the cell divides in half to make 2 cells. They each get half of the DNA. So they each have the normal amount of DNA. If we want to compare that to meiosis, an easy way to think about meiosis is, in meiosis, the cell replicates its DNA, same as mitosis, but then the cell divides twice. So after the first division, each cell has the same amount of DNA as the original cell. After the second division, since the cell doesn't replicate its DNA again, the cells only have half as much DNA as the parent cell that produced them. So that's an easy way to think about it. Now, when we look at meiosis and we look at the stages in meiosis, there's a couple of things we want to keep in mind. Usually we don't talk about prometaphase in meiosis. We just have prophase and metaphase. Everything that happened in prophase in mitosis, pretty much it still all happens, but we just say that it's all part of prophase. Secondly, in meiosis, since there's 2 cell divisions, we have 2 of the different stages. So we have meiosis 1, which is the first meiotic division, and we have prophase 1. This shows early and middle prophase, which is kind of equivalent to prophase and prometaphase, but they haven't named them that. And then we have metaphase 1, anaphase 1, and telophase 1. And then in meiosis 2, or it's sometimes called the second meiotic division, we have prophase 2, metaphase 2, anaphase 2, and finally telophase 2. And notice telophase 1 happens with cytokinesis. Telophase 2 happens with cytokinesis. So we end up getting 4 daughter cells from 1 cell that started the process. Now, notice that there is an interphase before meiosis 1. The interphase isn't numbered because there's not usually an interphase before meiosis 2. So usually the interphase before meiosis starts is just called plain old interphase. You might see it called interphase 1, but usually it's just plain interphase. Now, except for interphase, if we're talking about the stages in mitosis and meiosis, and you saw a bunch of them on a multiple choice question, how could you tell whether the stage was from meiosis or mitosis? Well, except for interphase, notice that all the stages in meiosis have numbers with them, either 1 or 2 at the end, depending on whether they're part of the first meiotic division or the second meiotic division. In mitosis, if you go back and look at your notes from yesterday, the stages don't have any numbers associated with them. So prophase is just prophase. It's not prophase 1 or prophase 2. like in meiosis. So if we're looking at a bunch of stages and we're trying to just figure out real quickly whether they're in mitosis or meiosis, if they've got numbers, they're part of meiosis. If they don't have numbers, they're part of mitosis. So we want to keep that in mind. So if we look at prophase, notice that this is a cell that starts out with a 2n number equal to 4, or a diploid number equal to 4. There are 4 chromosomes. There's a long blue chromosome, there's a long red chromosome, there's a shorter blue chromosome, and a shorter red chromosome. Most of the time that we're talking about genetics, we're going to use blue, since that's the color for baby boys, to represent the genetic material from the father. And red, it's as close as we can get to pink, since that's the color for baby girls, to represent the DNA from the mother. Now, remember, back in interphase, during the S stage, or it's sometimes called synthesis, the cell replicated its DNA. So when we first see the chromosomes, what we actually see is we see pairs of sister chromatids connected through that part of the DNA called the centromere. So when we see these 8 sister chromatids, if we didn't know before, that tells us that there were 4 chromosomes in a diploid cell, or a 2n cell, if you prefer. Now, there is one difference between what we see in prophase of mitosis and prophase of meiosis. In mitosis, if we look at the pairs of sister chromatids, they're kind of scattered all around the area of the nucleus. There's no particular pattern. It's kind of like that children's game of pickup sticks, if you ever played that when you were a little kid. But in meiosis, we see something different. The pairs of sister chromatids from homologous chromosomes are close to each other. So that means the 2 blue sister chromatids are pretty close to the 2 red sister chromatids for the longer chromosome here. The 2 blue sister chromatids are pretty close to the pair of red sister chromatids for the shorter chromosomes here. Remember, homologous chromosomes are the same size. They have the same genes. We can't see here, but remember we said if we put a stain on them, they have similar banding patterns when they absorb those dyes. So when we look at prophase 1, what we see is, if we look at a blue pair of sister chromatids and a red pair of sister chromatids from homologous chromosomes, they're located near each other, and very soon after the beginning of prophase 1, these homologous chromosomes, remember, homologous chromosomes, same size, same genes, same pattern of absorbing dyes, these homologous chromosomes attach to each other. This process is called synapsis. So when they attach, we have 2 chromosomes, because the 2 blue sister chromatids are just 1 chromosome. The 2 red sister chromatids are just 1 chromosome. So we have 2 chromosomes, or if we want to count sister chromatids, we have 4 sister chromatids attached. So because we have 4 sister chromatids attached, this clump is sometimes called a tetrad. Tetra means 4. Or because we have 2 chromosomes, it's sometimes called a bivalent, and bi means 2. So one of the things we see in prophase 1 of meiosis is tetrads, or bivalents. And we didn't see those at all in meiosis. The pairs of sister chromatids stayed separate from each other. They didn't attach. There was no synapsis. So we want to keep that in mind. Now, because of this synapsis, we have something that happens in meiosis that didn't happen in mitosis. So here's our 2 red sister chromatids. So let's think about what happens to those in mitosis. So we want to keep in mind that they stay separate from the 2 blue sister chromatids during mitosis. You might remember I said that during mitosis, even though the chromosomes are coiled up, even though they're more compact than they are during the interphase between mitoses, that sometimes the tips of the chromosomes get tangled up. Well, there are enzymes, proteins, there are enzymes that come along and kind of snip out the tangles and then glue them back onto the ends of the sister chromatids. Now, sometimes these enzymes make a mistake, and when they snip out the tangle, they cut out the end of the left sister chromatid here, and they accidentally paste it onto the right sister chromatid. And the end of the right sister chromatid that was tangled that they cut out, they paste onto the left sister chromatid. Now, this doesn't really matter in mitosis, and it doesn't matter because of the fact that genetically, these two sister chromatids are identical. Remember, we said if the cell replicates its DNA accurately, the DNA in the left sister chromatid is exactly the same as the DNA in the right sister chromatid. But in synapsis, in meiosis, we're going to have a problem. Notice we have a blue sister chromatid that's next to a red sister chromatid. And if these get tangled up and those enzymes come in and they sniff out the tangles, and then after they snip out the tangles, they reattach them. If they reattach them to the wrong sister chromatid, we're going to have a sister chromatid that's mostly blue, but has a little red tip on it. And we're going to have another sister chromatid that's mostly red, but has a little blue tip on it. So this is going to create a different combination of genes. Because remember what we said a few days ago, you might not inherit the same instructions for proteins from your mother as you inherit from your father. So you could have a sister chromatid that you inherited from your mother, and it's got a few of the instructions that you actually inherited from your father. So this is a process called crossing over. So let's look at our chromosomes with the two sister chromatids again. And we're going to imagine that these chromosomes have two genes on them. Now, keep in mind, in real life, chromosomes have an average of about 1,000 genes. So one of the gene we're going to designate by the letter A. And maybe letter A is the gene for eye color. So maybe big A is the instructions. And remember, we said those different types of instructions are called alleles. Maybe big A is the allele for brown eyes. And lowercase a is the instructions for blue eyes. So we have different instructions on these two chromosomes. And maybe big B is the allele for type B blood. And little b is the allele for type O blood. So again, we have different instructions on these two sister chromatids. Now, if they get tangled up and if they get reattached to the wrong sister chromatid, notice that this creates some new genetic possibilities. Because if we look, what you inherit in your father's sperm cell or in your mother's egg cell is you inherit a complete chromosome. So notice before crossing over, if this was your father, you could only inherit big A along with big B. If this was your mother, you could only inherit little a with little b. But now you could inherit big A with big B with little b, little a with big B, or little a with little b. So there's more genetic possibilities. And this is something that's called recombination. Recombination is making new combinations of DNA. Here we're taking some DNA that a person inherited from their mother and it ends up stuck onto a chromosome they got from their father. Just a sperm fertilizing an egg is a type of recombination. But notice what this does is, if this is me, if this is the chromosome I got from my father and this is the chromosome I inherited from my mother, I could only make two types of sperm cells before crossing over. My sperm cells would either have the big A and big B in them or little a and little b. After crossing over, I can make 4 different types of sperm. So this means that there's more genetic combinations. Now, why is this important? Well, remember, we're looking at a type of sexual cell reproduction. In mitosis, cells that are made are exactly like the parent cell genetically, and they're exactly like each other. Now, when we look at any type of sexual reproduction, as far as we can figure out, one of the main advantages is more combinations of genes, more combinations of DNA. Why does this matter? Well, if you reproduce by mitosis, that's okay, as long as your environment never changes, as long as things are always exactly the same. Because since your offspring are the same as you genetically, they might not do so well in a different environment. But by mixing up the genes by different types of recombination, and crossing over is just one type, this gives us more genetic combinations in the next generation. So if the environment changes, there's more likely to be some individuals who can cope with that change. If everybody is genetically the same as their parents, not so much so. So crossing over happens during prophase, while the sister chromatids from the two homologous chromosomes are attached. So the two red sister chromatids and the two blue ones, and it always happens on the inside pair, because this outer blue sister chromatid isn't really next to this outer red one, so we don't see crossing over there. So metaphase, like in mitosis, is always the easiest stage to describe. In metaphase, the tetrads get to the middle of the cell. Now remember, in mitosis, we just had pairs of sister chromatids in here. Here, it's the whole tetrad. And notice they're showing on the tetrads, the red sister chromatids on the inside have a little tip of blue on them. The blue sister chromatids on the inside have a little tip of red. If we look at human chromosomes, except for the X and Y chromosome, crossing over happens about 4 times on every pair of chromosomes in the human nucleus. And because of crossing over, and because of the fact that you could pass down the chromosome from your mother for number 1, the chromosome from your father for number 2, because of all those combinations, a person can make about 90 million different eggs or 90 million different sperm. So this means it's very unlikely that you'll have 2 children that look exactly the same, unless they're identical twins. Does anybody know why identical twins are genetically the same? Does anyone know how identical twins are actually produced? Well, normally when a woman goes through the ovarian cycle each month, she releases 1 egg during ovulation. But in some cases, after that egg is fertilized by a sperm, before we get to 16 cells, so that 1 cell, after it's fertilized by a sperm, that fertilized egg is going to divide by mitosis. We get 2 cells. Remember what we've said about mitosis. It's asexual cell reproduction. So the 2 cells are genetically the same. They divide by mitosis. We get 4 cells. They divide by mitosis. We get 8 cells. And if something happens and those 8 cells break apart into 2 separate clumps, a clump of 5 cells and a clump of 3 cells, both of those clumps can develop into a complete fetus, a complete baby. So since they had the same DNA because they came from the same egg cell, and they also got the same DNA from the father because the same sperm cell fertilized that, genetically, these twins will be identical twins. They have exactly the same DNA. Now, notice that even though in most of their characteristics, identical twins are the same, identical twins don't have the same fingerprints. So if you're going to commit the perfect crime and you're going to frame your identical twin for it by leaving fingerprints behind, keep in mind, your fingerprints will be there, not your identical twins, because they're different. So what happens After metaphase 1, in anaphase 1, the homologous chromosomes separate. So basically what's on the left side of our cell moves to the left. What's on the right side of our cell moves to the right. And then in telophase 1, the homologous chromosomes actually get to opposite ends of the cell. The cell rebuilds the nuclear envelope around them sometimes. Sometimes it doesn't even bother. But notice that when they get here and cleavage completely divides these cells, notice we can tell that this is an animal cell in meiosis if we weren't sure, because remember, when a plant cell divides, cytokinesis happens by a cell plate being built in the middle of the cell. In animal cells, the cleavage furrow pinches in from the outside. So even if this cleavage site wasn't labeled, we could tell that this is a cleavage furrow and we would know that this was an animal cell. But notice that each cell only has 2 chromosomes. Now the chromosomes are still made of 2 sister chromatids, but each cell only has 2 chromosomes. Well, since we started with 4 chromosomes back in prophase, now we have 2, so we're going from a diploid cell to a haploid cell, or if you would prefer, from a 2N cell to a 1N cell. Remember, diploid and 2N mean the same thing, haploid and 1N mean the same thing. So what do we see in prophase 2? We see exactly the same thing that we saw at the end of the first division. So over here on the left side, we have our 2 chromosomes that are mostly chromosomes we inherited from our mom, but with little parts that we inherited from the father due to crossing over. And in this cell, we have mostly chromosomes that we inherited from our father, but again, some small parts that we inherited from our mother due to crossing over. Now notice in real life, we don't necessarily make eggs or sperm that have all the chromosomes from the mother or the father in the same cell. So prophase looks the same as it did at the end of telophase. Notice we never have any new sister chromatids because we only make new sister chromatids. We only replicate the DNA during interphase, and there is no interphase before metaphase 2. We just go straight from telophase 1 and cytokinesis to prophase 2. So we have 2 different cells now. Now, in these 2 different cells, the chromosomes get moved into the middle. And notice this is very similar to what happened in mitosis, except we only have 2 chromosomes. If this was mitosis for this cell, we would have all 4 of the pairs of sister chromatids in the middle. So the chromosomes, which are still pairs of sister chromatids, move into the middle of the cell. Then the sister chromatids split apart and move to opposite ends of the cell. And then finally, in telophase 2, cytokinesis happens again. The cell rebuilds the nuclear envelope. And notice that each cell just has 2 chromosomes. They have one of the bigger pair of chromosomes up here, one of the smaller pair of chromosomes. So we have a cell that started out with 4 chromosomes. Now we have 2. So at the end, we have 4 haploid cells, our 1N cells. They each have half as many chromosomes as the parent did. And this is okay, because if these are sperm cells, they're going to have to fertilize an egg cell. And for a person, a sperm cell would have 23 chromosomes, an egg cell would have 23. So when those combine, we're back to 46 chromosomes, the number in a normal human cell. So when we look at meiosis, meiosis happens in organisms that reproduce sexually, because organisms that reproduce sexually have a male and a female parent, and they're both going to contribute the same amount of genetic material. So by cutting the number of chromosomes in an egg or a sperm or a spore or a pollen grain, by cutting that number down to half of the normal number, the haploid number or the 1N number, That means that, for instance, human cells always have 46 chromosomes. If we made our sperm or egg by mitosis, that would mean a sperm had 46 chromosomes and an egg had 46 chromosomes. So the next generation would have 92 chromosomes instead of 46. And then they would make sperm that had 92 chromosomes and eggs that had 92. And the next generation after that would have 184 chromosomes. So this keeps the number of chromosomes constant, and it also kind of shuffles the genes around, so we get different combinations. And as I say, the different combinations are useful because genetic variability can help organisms adapt to new environments. If we look at genetic variability, we don't always understand why it's important. But when we learn enough about it, usually we can see some of the importances. If we look at people, for instance, there's 4 different blood types. You could be A, you could be type B blood, you could be type AB, or you could be type O. And for many years, we didn't know why these different blood types were important. But we've now learned that certain blood types, I should say maybe people who have certain blood types, are more resistant to certain diseases. And with COVID-19, there's even some evidence that people with type O blood are less likely to get infected and less likely to die from COVID-19 than people with type A blood. Now, it's not a very big difference. So if you have type A blood, don't run out and panic. You're not doomed. But there are differences with things like cholera and typhoid fever and some other diseases as well. So for many years, we didn't know, why all the different blood types. It doesn't seem like they matter. Well, it turns out they do matter. They might not matter as much now as they did, say, 50,000 years ago, when people lived in a very restricted geographic environment, and you might never move very far from where you were born, so you were always subjected to kind of the same diseases as your parents were and your grandparents. So, then it might have been a big deal if you had type O blood, because that might have protected you against the diseases that were common in that small geographic area. But now that people can get on a jet plane and be any place in the world, these are probably a little bit less significant. Now, some genetic differences, as we'll see when we go along, are much more obviously important. So let's look at people, and let's look at the difference between males and females when they go through meiosis. So let's look at males first, because males are simpler. So the cell that kind of begins the process of making a sperm is called a primary spermatocyte. Primary means first, so there are secondary spermatocytes. And a primary spermatocyte goes through a process called spermatogenesis. So anytime you hear a biological process named Genesis, and there are a lot of them, think of the Bible and the book of Genesis. This is how things begin according to the Bible. So in spermatogenesis, this is how sperm began, or this is how your body makes sperm, is another way to think about it. So in spermatogenesis, what we see is the primary spermatocyte divides, and in the first division of meiosis, we get secondary spermatocytes, and they divide, and we get some cells called spermatids. And spermatids eventually become the cells that we call sperm. Now, besides how this happens, another thing that we have to think about if we're going to compare males and females is the fact that this process begins in boys when they hit puberty. And then once they hit puberty, there are millions, in a few cases, only hundreds of thousands of sperm produced every day. As men get older, they tend to make less sperm and they make more defective sperm. But as far as we can tell, pretty much till the day they die, men are making sperm. Now, let's look at what happens in females. In females, the first cell that's produced is a primary oocyte. Oo means egg. So this is a process called oogenesis. So again, the beginning of egg cells, or how you make egg cells. Now, when the primary oocyte divides, one of the secondary oocytes gets most of the cytoplasm and most of the organelles. And the second one gets half of the genetic material, so the DNA is distributed equally, but it doesn't get very much cytoplasm or very many organelles. And this is called a polar body. Now, the secondary oocyte and the polar body divide again in the second division of meiosis. And again, one cell from that secondary oocyte gets most of the cytoplasm, and this becomes the ovum, or the egg. The polar body divides and makes two polar bodies. The secondary oocyte that was the largest makes another polar body. So in females, only one of these cells becomes an egg, the ovum. The other three cells usually deteriorate. I say usually because every once in a while they actually stick to the ovum, to the egg cell. And you've probably heard of identical twins, and you may have heard of fraternal twins, which aren't genetically identical. But you can also have something called polar twins, where this polar body sticks to the ovum, and the ovum is fertilized by a sperm, and the polar body is fertilized by a sperm. So one difference is the number of cells that are produced in spermatogenesis versus oogenesis. Here we get 4 cells from every sperm that starts the process. Here we get 1 egg from every cell that starts the process. And actually we don't even get that because of the way that eggs are made in females, at least in mammals. This isn't true for all animals, so we want to kind of qualify what we're talking about here. It's true for people, and it's true for other mammals. So let's think about a person first. So males start making primary spermatocytes when they hit puberty. For females, what we see is that all the primary oocytes that a female will ever make are made by about 8 weeks before she's born. Now, keep in mind, that's thinking about the normal gestation period, which is about 40 weeks. So by about 32 weeks after the egg is fertilized, all the primary oocytes in a woman's ovary are made. Now, they don't do very much. If we go back to meiosis, all these primary oocytes start dividing by meiosis, but they stop in prophase 1. So they really only get to the first stage. They don't get very far at all. But that's all the egg cells that she'll ever make. Now, when a girl hits puberty, what happens is every month of the ovarian cycle, a bunch of these primary oocytes start to go through the other stages of meiosis. They don't get very far. By the time they finish telophase 1, they're actually being released in ovulation. And usually only one of those possible cells is released during ovulation. So that's why we said a minute ago, one egg cell is released in ovulation, it's fertilized by one sperm, and then it splits apart into 2 clumps of cells. That's identical twins. So how do you get fraternal twins? Now notice, identical twins have the same DNA, so they're both boys or both girls. And you can have identical triplets or something like that. If we look at fraternal twins, in fraternal twins, when a woman ovulates, she releases more than 1 egg. And each egg is fertilized by a separate sperm. So each egg cell has a different combination of DNA from the mother. Each sperm cell has a different combination of sperm from the father. So this means that when we look at fraternal twins, one could be a boy and one could be a girl. But genetically, fraternal twins are only as similar as normal siblings are. Now, there have been some studies showing that fraternal twins maybe tend to resemble each other a little bit more, not because of genetics, but because of environment. They're both in the mother's uterus attached to a placenta at the same time. And when they're born into the family after birth, they're born. So the family's socioeconomic conditions are likely to be similar. So there is some evidence that fraternal twins are a little bit more similar than identical twins. So when we look at twins, there are actually about a dozen types of twins. So we want to keep that in mind. So yesterday we talked about mitosis, which is a type of cell division that happens in your body. Today we've been talking about meiosis. So let's think about comparing these. So here we have a parental cell, kind of similar to that one we had up above. We have a long blue chromosome, a shorter blue chromosome, a long red chromosome, a shorter red chromosome. So notice the 2n number would be 4, or we could also say the diploid number is 4. And the chromosomes are replicated, and that's what we see during prophase of mitosis. Now, it's actually hard to see, but these replicated chromosomes are made of 2 sister chromatids. So the chromosomes line up in the center of the cell during metaphase. And during anaphase, they're separated into different cells. And then telophase and cytokinesis is what actually divides these up. Notice that the 2 daughter cells that we make each have one of the long red chromosomes, one of the short red chromosomes, one of the long blue chromosomes, one of the short blue chromosomes. So genetically, they're identical to the parent cell. So in mitosis, all the daughter cells we make are genetically identical, and they're identical to each other and to the parent cell. Now on the right side, it shows meiosis. We replicate the chromosomes, but the difference is we have synapsis. So instead of having the long red chromosome separate from the long blue chromosome going through mitosis, they clump together to form a tetrad. And similarly, the short red chromosomes and the short blue chromosomes clump together to form a tetrad. So in metaphase 1, this tetrad is what moves into the middle of the cell. Then half of the chromosomes, half of each tetrad, if you want to think of it that way, half of each tetrad goes into one daughter cell, half goes into the other daughter cell. Then we're going to go through something very similar to mitosis. For these 2 daughter cells, the chromosomes are going to move into the cell, middle of the cell, and get separated. So we end up with 4 cells that are each haploid, or 1n. And notice they've each got one long chromosome. It might be red, it might be blue, it might be mostly red with some blue, mostly blue with some red because of crossing over. And they've each got one short chromosome. So if we're looking at a type of organism that only has 4 chromosomes, these are going to be the eggs or the sperm. And when they meet up with their opposite, we're going to be back to 2 long chromosomes, 2 short chromosomes. So mitosis and meiosis are quite different. And for the upcoming test, when we have this, we want to keep in mind, this test won't be next Monday. This test will be the Monday after. But when we get to this point, you want to make sure that you know the difference between mitosis and meiosis. So let's look real briefly at the next homework assignment. And this is just posted today. Like usual, there's a PDF version if you have trouble seeing the chart. But basically, this homework assignment is a comparison of mitosis and meiosis. So most of the answers are listed for you. You know, what type of cells does this process occur in? Somatic cells are the cells that make gametes. It's the chromosome number 1N or 2N. So most of these numbers are down here. So this is due next Tuesday. But keep in mind, before next Tuesday, on Monday, we will have our metabolism test. The metabolism study guide is online under this week. There's also a homework assignment due Monday, which is the metabolism homework. There's also this homework, which is due Tuesday. So keep that in mind. Now, there's one more thing that we need to think about before we leave. And this is that Monday is the last day to withdraw from class without getting a grade. If you withdraw on Monday, you get a W. If you withdraw after Monday, well, you can't withdraw after Monday. If you stay in the class past Monday, you get a grade. So you should be able to go online and you should be able to see your grades. I think your grades are in the upper right-hand corner of your page. If you want to meet with me, I'll check, but I'm pretty sure I have it set so that you can see your average for everything we've done in the class. If you want to stay in the class or you think you do, but you're not sure, send me an email. And at least tomorrow, I know I have some open time and we could meet and discuss this in a Zoom meeting, or you can just send me an email and I can tell you what your average is. What I can't tell you is whether you should stay in the class or drop. Sometimes people ask me to predict what they're going to get on the last tests and the last assignments. I mean, the best prediction for what you're going to do on the remaining assignments is whatever you've done on the previous assignments, I would say, unless you know that your work schedule has changed or you've just been goofing off and haven't studied much and now you're going to actually study. You should probably think about whatever your grade is now and if you would be happy if you ended up with that grade, because your grade could still go up. We have the metabolism test next Monday and we have homework assignments. And remember what I said, we're going to have lots of homework assignments. We still have 4 more, 2 next week and 2 the last week. And then the last week on Monday, we have the test over mitosis and meiosis and genetics. And then our final exam is over DNA. So we really have about 40 or 50% of the points left. So your average could change quite a bit. But, you know, you probably have a better idea of how you could, how you're going to do at the end of the class than I do. So if you want to discuss your grade or anything, let me know. And like I say, I know I have some times I could meet tomorrow. I'm not so sure about Saturday or Sunday, but I may. If you just want to talk about the metabolism homework or the mitosis and meiosis homework or the metabolism study guide. So any other questions? Okay, if not, I will let you go. I'll double check and make sure. But like I say, I think I have the grade book set so that you can see your final averages right now. But I'll double check and see if that's the case. And if not, I'll try to change it. So you can go ahead and go if you would like, if you don't have any more questions.